Dat we live zijn. En zo'n ding. Dan geef ik een berichtje terug. Dit in wordt.
Vastgeklikt. Dus moet een tweede eraan gaan worden.
Een 3 met en zo?
Ja, wij hebben nog een verlengkabel hier gestoken. Maar...
Kijk, dan ga ik zo werken.
Ja, ik ben anders een tweede hier gaan vastklikken en dan kan het goed.
Ja. Misschien wat. Structureren buiten zo'n dagen. Ja, snap je. Zo. Good evening, everyone. Today, course number 6 in the bioinformatics, about structural bioinformatics. So I've uploaded all PowerPoints, which you can find in modules. So you'll see that there's a total of 11 chapters. First one is just a small introduction, and then the last part here, number 12, is actually already an example exam, closed book, open book. And also I think many of you will like this. There's also an entire course in Dutch, which is like 95% matching the PowerPoints. So last year I just translated all the PowerPoints, so normally for next year I hope to also translate the entire course, but you can already, I think, all of you are Dutch speaking, so you can use this if you want to study with a course. But as I told you, the PowerPoint, the slides are like 95% the same. Maybe here and there a new tool or something, a new figure that has been introduced, but for the rest it's more or less the same. On the homepage I also try to divide over the weeks what I think is doable for you. So here you see all the chapters again, and what you see is, this is what I will discuss this evening in contact moment 1. This is actually all the basics, and this will also be like half of the questions on the closed book exam. So the closed book exam counts for 50%, and the open book for you also for 50%. The students during the day also have a project assignment for 10%, but that's not for you. Then normally you have to read a paper of a structure and present that. Well, of course, we don't have time for you to all present the structure. So for you it will be 50% for the open book exam. This is also a skills exam with some questions on databases, on structures, and then visualization. So you will definitely get 2 visualizations. A global overview of a certain structure and a detailed visualization of a structure. It's always the same each year. For the closed book, half of the questions will be of these 3 chapters, because these are actually the most important ones, and half of the questions will be from the other chapters. So this is obviously everything you need to know about protein structure and about nucleic acid structure, but probably you all have biological background. I hope you already know what amino acids are, what nucleotides are, so that won't be that new for you. And an experimental structure determination is actually all of the structures that you will visualize during the coming weeks are deposited in the protein database. So this is actually one of the databases discussed in the database chapter. But how do we get these structures? These coordinates is actually just x, y, z coordinates of each of the atoms. How do we get these? This is discussed in experimental structure determination. So there are 3 methods: X-ray crystallography, NMR, and electron microscopy, and that will be discussed in this chapter. And this is also what I will discuss this evening. Let's say half an hour for each part, and in the last half hour, or even hopefully a bit more, I want to give a workshop about how you can use PyMol. So the molecular visualization software that you have to use is PyMol. Some of you already know PyMol. So I will give you an introduction again, so that will make it easy for you for the coming weeks to start using PyMol and interpreting, visualizing structures. So this is for this evening, and then the next part, so next contact moment are 4 smaller chapters, and for the last contact moment again 4 smaller chapters. Databases is quite long in terms of PowerPoints, and I can actually tell you days about this. But one of them, for example, is NCBI PubMed, so it will depend how good you already know how to work with this one. What is interesting, actually, today, earlier, I uploaded some recordings here. So I don't know, for the R course I already uploaded some recordings. Now you will see here, if you will go to Panopto recordings, that quite a lot of movies I already have from the day courses, not yet from all the chapters, so by next year I hope to have also the missing parts. But you will see, for example, you will have 2 movies on protein structure. For each of chapter 4 you will have a part. So X-ray crystallography, NMR, electron microscopy. So this is, on average, I think more or less like half an hour. I think the smallest one is 10 minutes and the largest one is 1 hour, but most of them are like 25 minutes, 30 minutes. And then on databases you will see that there are several ones, I think 5 movies. So it will depend how good you already know these databases. You can have a look at the movie, because of course I can't talk only about databases next time for 2 hours. So if you want to know more, you can just have a look at these movies. For example, it's also included as GenBank, reference sequences on NCBI, so I don't know how familiar, maybe in another course you already saw this. But actually the most important one then is actually the PDB database. So that's a separate movie here, about 40 minutes on the PDB database. So this is definitely one you should have a look at. The other ones I could say is optional, you could just also study from the course. Well, this one is an interesting one to have a look again on how you can use the PDB database. So, and then you will see also for the other ones, chapter interactions and on prediction, there are also movies available. So let me go back to the modules and start with the introduction. And this is actually just situating where structural bioinformatics is actually. So there's a very good book on structural bioinformatics, already a bit outdated. That's why I try to include new tools, new servers, for example, for structure prediction. I try to include these from actually the latest literature. But this is definitely a good book to start with. And actually, many of the chapters in the course are actually based on this book. So this is again the overview. So this is not for you presenting a published structure, but you do have to spend quite some time practicing molecular visualization. And for the rest, actually, it's more studying this course. I think for all the courses you had so far, this is probably the one you will have to study most for, and maybe less practice. So here the focus is on the molecular visualization. So what is structural bioinformatics? I think you already now know the difference between wet lab and dry lab. And actually what we do is, in bioinformatics, we can split it up in 3 components. Let's say the theory and the methods, the algorithms that are being developed. Then we have the applications. What can we do with all these methods that are developed? And then of course, an important part is the data. Sequences, but can also be mass spec data, next generation sequencing data, but also structural data. And this is actually what we'll do in structural bioinformatics. There are quite a lot of algorithms, methods that we'll use. We use those on biological structural data, but also we can include actually sequence data, for example, for comparing sequences. A sequence or an amino acid, for example, that is highly conserved is most likely more important and has an important function in the structure, for example, for stabilizing the structure could be one example. Biological structure, you can see that at different scales. So you can just have a look at molecules, larger complexes of molecules, but also then on a higher level, organelles, cells, and of course also complete organisms. I think that's obvious if you want to do molecular visualization, that will focus mainly on this level. So molecules and larger complexes, although of course with microscopy, from microscopy, you can have even larger things you can visualize. And in this course, the focus will actually be on proteins. We will have a look at DNA, RNA, ligands, and complexes, but the main focus will be on proteins. So that's for the introduction. So then let's have a look at the structural features and the fundamentals of protein structure. So proteins are, of course, very important structures, and actually this is what I always cite is if you want to study the function of a protein, you actually have to have a look and understand the structure. So protein structure leads to protein function. If you can understand the structure and see how it works, then you can actually know how a protein functions. So this is why actually structural bioinformatics is important. What is the difference between, for example, if we compare protein structures, actually it's nothing more than just chemical groups that are at specific places in the three-dimensional space. And often you have, like of all your protein, you have certain spots on the protein that actually are very important. For example, in the case of enzymes, that actually those active sites, we call them then, are important for the chemical reactions. What is the first structure that was known actually already in 1958 was the protein structure of myoglobin, and shortly after that Perix also determined the structure of hemoglobin. They have to think in 1958, 1959 they had to work with this. They didn't have a laptop with Pymol to visualize actually, so they have to work with these sticks and balls to try to see how the structure looked like representing the atoms. And they actually won the Nobel Prize in Chemistry in 1962 for the structure of these proteins. And you will see actually that in the rest of the course, you will see lots of Nobel Prize winners passing by because of the thing they actually invented. For example, also X-ray crystallography, Röntgen is, for example, also very important. The rest of the chapter is actually, we can divide in these 4 parts, depending on which level you want to have a look at the protein structure. And actually the first level is just the primary structure, the amino acids, the building blocks, the basic building blocks. The second level then are the secondary structures. I think you're already familiar with beta sheets and alpha helices. Yes? The tertiary structure is, of course, then the protein as a whole, and then you can have even another level. The quaternary structure is then actually where you have complexes of proteins. It can be the same molecules, different molecules, depending if you have homodimer, for example, or heterodimers, that would have same or different molecules. So this is not to describe physical laws, but actually just it makes it easier to understand the protein. Okay, and then the basic building blocks are the amino acids. So I think you also know that an amino acid is built like this, with an alpha carbon atom. And then you have these two, and here you will have your peptide bonds, then you have your hydrogen, and then this is your variable group, your side chain actually, as we call it. And changing this side chain determines then which of the 20 amino acids you will have. So these are the 20 amino acids. You have to know these amino acids. What do you have to know? Of course, you have to know all 20 of them, full name, 3-letter code, 1-letter code. And also very important in structural bioinformatics, you have to know the biochemical properties. You have to know, is it a charged amino acid? Is it hydrophilic? Is it hydrophobic? That's very important. What I will not ask you is, draw me the structure. That I won't ask you, okay? But it's important if you look at the structure, or if you look at a certain motif, for example, an active site, that you at least know, for example, that Y here is a tyrosine, that W is a tryptophan, and things like that. That's very important that you know that. And that's always also a question on the exam. Always. You will always get one question about amino acids. Okay, and then of course, for proteins, you can also have post-translational modifications, which can be important for the function of the protein. So you have glycosylation sites, phosphorylation sites, methylation sites, for example, depending on which group is added on top of the protein or on the amino acids. You know there are 20 amino acids in nature, but actually we have some more. For example, a variant of cysteine is actually selenocysteine, where you actually have a selenium atom here instead of the sulfate here. So this is now what we call the 21st amino acid. And then you also have pyrrolysine, is actually then the 22nd amino acid. And some proteins of metallogenic bacteria can actually incorporate this in the proteins. This here is, I think, a nice figure to show you the different properties. So here you see the hydrophobic group, then this is the polar, the hydrophilic, and then these are the charged ones. And these are just some slides if you want to train yourself what the properties actually are. And then some special amino acids that can play a special role also in the structure. Is, for example, proline is actually not an amino acid but an amino acid. Why? Because actually here it is cyclic. So this actually limits its flexibility. It also will have a role where you can find proline. If you would have a look at an amino acids sequence, often just by looking at it, you can deduce certain features. For example, if there would be, let's say, a transmembrane domain, I think by the end of the course, just by looking at the amino acid sequence, you might be able to say, probably here there is a transmembrane domain. Just by looking at the sequence. Why? Because you have lots of hydrophobic residues. Prolines, for example, lysines, they often we find them, for example, in loop regions. Because you need a short turn, and actually proline and glycine makes it more possible actually to have such a short turn actually. So glycine is another one, has no side chain, and this then has an influence on the symmetry actually, because this is then symmetric while the other ones are non-symmetric. And if they are non-symmetric, this means actually you can have two forms. Probably you also know that you have L forms and D forms depending on how you see it, and then you can have a look at this figure, try to see actually how it is. So if you just turn this over, you will see that it's actually not the same. Well, here it is in the front. If you would turn it, the R group would actually be in the back end. And then, of course, you probably already know that these amino acids then form peptide bonds. If they are short, we call them a peptide. If they are longer, we call them a polypeptide. And polypeptides are then forming the proteins actually. The central part here is what we call the backbone. So these are the alpha carbon atoms and then the residues involved in the peptide bonds. So if I refer to in the rest of the course to the backbone, this is actually what I will be referring to. And then, of course, you have the beginning and the end is the N-terminal part, the C-terminal part, and then you have typically the side chains showing, so the R groups. And then maybe this is new for you, some characteristics, some features about these structures. Actually, you have to see this as a plane. Let's say a piece of paper. So if I would have some papers here, yes. So this is actually the alpha carbon, and this is then where you have the bonds, right? And you should actually, if you want to visualize that, it's just these, and actually these can just rotate like this. So this means you can have a different angle here and there. We call this the phi angle and the psi angle, or also the dihedral angles. And this actually limits the spherical hindrance between the side chains. So not all angles are possible if we look at a protein. And this is actually a feature that we can use to also have a look at the quality of a structure. If for a protein we can actually make a plot of all these angles, and then we know if the structure is reliable or not. This is something that will be important if you want to predict the structure for yourself. You can use this to see how reliable the structure is, if the quality is good or not. So that is about the primary structure. Then the secondary structure is actually the local three-dimensional structure. So then typically we have alpha helices and beta strands represented like this. So this is actually what we call the sticks method or stick representation, where we will visualize the structures in PyMol. You can actually choose how you want to visualize them. You can choose sticks. This is what we will call cartoon. So this will be an arrow or a helix. So arrows then will be strands. helices will obviously be alpha helices. But you can either choose. If you have a quite complex structure, of course, if you would visualize everything as fixed, it will be very difficult to deduce anything or actually look at the function of the structure. So then, for example, in most cases you will see that we'll try, for example, to use the cartoon representation. But there are also other ones, as we'll see later on. You, for example, have spheres, where you have like balls, large balls, or you can also have surface. I always refer to that as like a moon landscape. Where, for example, if you have like a bit of a hole or a cleft, that's typically, for example, an active site. And this is actually such a plot of these angles, these VMC angles. It's what we call a Ramachandran plot, developed by the person who came up with the idea. And each block you see here, also here, each dot, represents an amino acid. And then here you see the angles. So this is 0 degrees, and then you can have either plus 180 degrees or minus 180 degrees for the one angle and then for the second angle. And based on that, then we can decide, okay, these two angles, so this is, for example, the amino acid. What we see is, if we do this for the structures, we'll see that if an amino acid is in an alpha helix, then you will see it in this region, which means this is typically the angle that you will have in an alpha helix. If the amino acid is in a beta sheet, then it should be in this region. And then, of course, it depends on the type of tool you use, that the region is like a bit different. But normally, this is where you have the amino acids for the alpha helices, this is the region for the amino acids for the beta sheets, which means if it is outside this region, actually this is not possible. Normally you can't have those angles, which means the position of that amino acid is very doubtful. Okay, so this looks quite good. You only have for the entire protein like 3 red ones, which means only 3 amino acids. If you would have, let's say, half of quite a lot, this means actually this structure is unreliable. There's actually a chapter also discussing that, as you will see later on in the course. So this is the Ramachandran plot. And then a bit more in detail, you will see we'll have slides on these secondary structures describing the typical features of, for example, the alpha helices, how many amino acids you will have, and then also important, which bonds are being formed to stabilize. And you will see that also here in the secondary level, but also on the tertiary level, that hydrogen bonding, for example, is very important. So these are the dotted lines that we see here between an oxygen and a hydrogen. And this is just to show you actually that this is right-handed and this is left-handed. I always tell the students you have to be on the scroll on the slide. If you sit on top here, then you would move like that. Okay, so this is on the right. If you sit on the top here and you would slide down, you would go to the left. This is the easiest way to remember that. So left-handed and right-handed. So the helices in protein structures are right-handed helices. And then you will see a number of features. There are other forms of helices, probably, maybe this is less known. For example, you can have a 310 helix, and you also can have a P helix, as you can see here. The difference is how the bonds are being formed. So, and this is then explained here, the bonding between the number of positions an amino acid is on. And then you have to count them actually, how far it reaches. So for example, here for the rare P helix, you will see it has 4.4 residues for one turn in the helix, and then the hydrogen bonding is formed between each residue and the residue 5 positions further. And this is then indicated here with the arrows. Now the second one is the beta strand. So a beta strand is 1. If you have multiple beta strands, they can form a beta sheet. Often students miss and they say they only see one, and they say if I would ask, well, now there are two, but let's say if there's one, and I would ask which secondary structure, say it's a beta sheet. No, it's a beta strand, you only have one. So that's important to remember. And then you can have different forms. Because typically this is how it looks like. If you have two, they can be either parallel or anti-parallel. And this is actually shown further here, depending on the direction. So parallel means in the same direction, anti-parallel means in the opposite direction pointing. And then normally you see here how the hydrogen are being linked to the oxygens. So this is the same as with the helices, but now in the beta sheets. And then you see here also the difference, how it is linked between parallel and anti-parallel beta strands. And actually the third local structure you can have on the secondary level are, of course, you have to connect all of these helices and these strands. How do we connect these with loops or coils? These can be subdivided or subclassified based on the length of the loop. So for example, the shortest one is of course the hairpin loop, shown here. So here you have two strands with a short hairpin loop, typically 4 to 5 residues. A bit longer, 6 to 16 long amino acids is the omega loop. And why the omega loop? Because they actually form like the Greek letter omega, like this. This is why we call them omega loop. And then you can have even longer loops. This is then the extended loop or the random coil, as is for example shown here. That's also possible. So that's for the secondary level, and then actually the global 3D structure, you'll see you have actually similar bonds. For example, the hydrogen bonds there are very important. But also on the tertiary level, the side chains of the amino acids play a more important role. For example, if they are charged, they could play a role. For example, hydrophobic, I don't know, where would you think the hydrophobic residues would be? On the inside of the protein or on the outside of the protein? If they are hydrophobic, where do you think?
I would say on the inside.
On the inside, of course. The hydrophilic would be more on the outside, because by a protein you can have it in, let's say, a water solution. But if you would have them on the outside, the hydrophobic ones, that of course would be a problem. Unless, of course, that could be stabilized, it would have another protein coming into contact, and then you would have two hydrophobic surfaces. But in most cases, you will have it on the inside. Same if you would have hydrophilic, if you can compensate that on the inside, of course it's not a problem. Also with charged residues, if you would have a charged residue on the inside, minus. If you would have a positive one, of course, again no problem, also on the surface. You would have on the surface charged residues, normally if they interact on the opposite protein or other molecule, again that needs to be compensated. So this is more or less, if you look in three-dimensional structure, how you should think. Hydrophilic, hydrophobic and charged. So protein, of course, are being formed amino acid by amino acid, and then actually they are folded into these 3D structure, and it actually occurs in, let's say, the nanosecond, millisecond range, as you can see here. So this is an illustration of how this folding actually occurs. And then we have the protein fold, is actually the final three-dimensional tertiary structure of the protein. Now we can, in a protein, you can have, of course, multiple domains. So for example, if you would have a protein of, let's say, 1000 amino acids, probably that will be divided in several domains. And these domains might, for example, be connected by linkers, loop regions. And later on you will see that you can actually have tools to find these domains and these motifs. Some of them are structures that we can see reappearing in different kinds of proteins. And this is actually what this part is about super secondary structures. So for example, different proteins can actually have this helix-turn helix motif, which is what we call an alpha topology. Why? Because we only have alpha secondary structures in it. Another one is what you can have is a 4 helix bundle, shown here. So this can be parallel here, or again anti-parallel, and then this is probably, if you would look on the top, how it would look like, and this is what I just discussed. You have a hydrophobic surface, for example, on the inside, and a hydrophilic surface on the outside. Then same as with the alpha, you can have the beta hairpin, we call it, which is then a beta topology, shown here. You can have a Greek key topology. This is a special one, but we see that also quite a lot in nature. What is the Greek key topology? You actually have 3 next to each other, anti-parallel, and then the last one actually makes a long loop with another one anti-parallel next to the first one. This is typically a feature of the Greek key topology. Then we also have the beta sandwich. Why? Because these 2 plates actually are on top of each other like in a sandwich. And for example, what is very known is, I think, is an antibody. I think this is the typical picture you see for an antibody. Yes, but actually, do you know how it is composed? This is actually how it looks like. So this is one beta topology. You see here the strands, and this part, this structure, is actually shown here. So, and then you can see each of these structures inside actually forms this antibody. So these are then these supersecondary structures that actually form or construct this antibody. And then another one, for example, is a beta barrel, and there's actually quite a nice exercise for in the workshop of molecular visualization about this beta barrel. This is typically, for example, if you have a porin, or you need in your cells, you have to transport molecules in and outside of cells. It's like here in the classroom, if you want to go inside and outside, we need a door. So also in our cells, we need doors, let's say. So these are typically these lipid layers. And then we have actually these kind of proteins inside of that. So this is then hydrophobic on this part. Now on the inside, you can actually have molecules going inside and outside. And there's actually a very nice example of that in molecular visualization. You will see that there's one loop here that actually will make sure that the pore is open or closed. So one loop can actually just change the structure and close or open this pore, changing actually if smaller molecules can go inside or outside your cell. This is actually how it works in nature. That's, I think, a very nice example of what we call a transmembrane protein. And then here another one is the Rossmann fold, then is a combined topology, where you have both alpha and beta secondary structures. And another one is the horseshoe topology. There's also an example of that. It might seem quite amazing that you can have this topology, but you will see there's also another example in the visualization workshop where you can have typically this structure where you have the alpha helices here on the outside and then the strands on the inside. A very nice structure, and of course it looks like a horseshoe. That's why we call it the horseshoe topology. So, what I already told you, hydrophobic effects, you can have charge and polar residues. Buried polar residues, of course, can also have hydrogen bonds with other polar residues, again for stabilizing the structure. And then, of course, you can have the pockets. So, pockets are then typically buried polar residues, for example. But you can also have hydrophobic pockets, and this then will allow other molecules to actually go into that pocket, like if you again refer to a door, you have a keyhole, you have to fit in the key. For these molecules, it's actually also the same how they can interact. You have a pocket, and then another part of the structure can actually go inside that and then make contact, and for example, have a reaction in the case of enzymes. what is important to tell. Yes, another kind of bond that I haven't talked about is the disulfide bonds. Of course, it depends which amino acids you have, because for a disulfide bond, you need then these thiol groups here, so you can of course only have it with cysteine residues. Now, important to know, that doesn't mean if you see cysteines in a structure and a sequence that they necessarily form disulfide bonds. That's important to remember. Okay, and then what do I want to tell more about that? Then you have special motifs. You can also have covalent interactions. For example, in this case you see here with zinc, but you can also have it with other metals, for example iron and other ones. We can also divide the proteins in globular ones, membrane proteins, and fibrous proteins, depending on which type of molecule it is. In most cases, we will study the globular proteins, of course. And there will be a few examples of membrane proteins, and I don't think we have an example of a collagen fibrous protein. If you would want to have a look at these kind of proteins, actually, this is always the kind of code you will see in the course. If you see a code like this is what we call the PDB code. If you use this code, you can directly visualize it. You'll see that later on during the course. Maybe I can quickly show it. Let's say if I would want to visualize that, I can just use fetch and grab it from PDB and then have a look. So in this case, 1 sp, 1 sp. And you see I can immediately fetch from the internet, from the PDB database, this structure. So it's important to know, if you see these codes, you can also quite quickly see how these proteins look like in Python. And then this further classification. And then the last part is in the quaternary structure. So here you see that this molecule is actually the same as this one. What is important then that you know the terms shown here. So proteins cannot function sometimes as a single chain, so one molecule, one monomer. Then of course you need multiple ones, so then we call them multimeric proteins, multimers, and this is actually what the quaternary structure is all about. And then it of course depends. In this case we have a homomeric protein, two of the same molecules, but it could also be different subunits. Then in that case it could be heteromeric protein that you have. But the interactions again, of course, will be the same as the previous level. So you can have again hydrogen bonds, you can have disulfide bonds, polar, non-polar interactions. So again the same as the previous level actually. And this is then the summary. So we have the amino acids in the sequence that can form peptide bonds. You can have a look at these angles, PMC angles, and plot that to have a look. Then you have the secondary level with the alpha helices and the beta sheets, and then different kind of loops to connect all of these. And depending on the composition, you can also have a level in between the super secondary structures, of which you have a few examples in the course. And you have the tertiary structure, and then if you have multiple molecules, you can also have a quaternary structure. And I think this is actually the first one that we'll visualize in Bible is the hemoglobin structure. So that's very quickly about this chapter. I don't know if that's a lot of new information or not. Still quite a lot.
It's
far away.
But I think it will come back soon.
Yeah. I think if you will have a look at it, it will come back. Okay, and then this is actually to show you now how many structures are experimentally determined. So I think the count now is probably, let's say, around 150,000. You can actually go to the PDB database later during the course, and there you will see the exact number, because on a daily basis new structures are being published. That doesn't mean that every protein that you would find in your research or in the lab, or that you can actually have a look at the structure. Why? Of course, might be that the structure is from mouse hemoglobin. And that doesn't mean that you can visualize human hemoglobin directly. But what you can do is you can sometimes predict the structure based on these 100,000 structures. Most of the domains are just being reused in nature as also a kind of building blocks. So if you would have a protein and there's no structure available in the PDB database, probably you will have these domains in your protein that will be similar proteins in a PDB database that has these domains, and then you can use those to build your own structure. That's possible, and that is what you will see in the prediction chapter actually. So it is perfectly possible for you to just predict and create a structure and visualize it in Python. Okay, so that's for the first part. Questions? No, not yet. Okay. And then the second chapter for the coming 2 weeks is again the same, but then DNA. So then lecture 3, of course. The chapter is actually chapter 2. Again, I thought, where is it? Here. So that is all about nucleic acids. So DNA structure, RNA structure. I think there will be a part here that's, at least the other students have problems with, that is quite difficult. Probably you know what the basic building blocks are of DNA, but then how the interactions are and how it is stabilized, that's often new, or to have a look at the details for the students. So probably you know Charles Darwin, you know Mendel, probably from high school with the flowers or the chickens, or typically these, I don't know if I have an example of that. Yes, here you do the crossing, dominant recessive, like that. But actually this is also an important guy, Michel here. What he actually did is he actually found the nucleic acids. He isolated a substance from cell nuclei from white blood cells, and he saw that it had a different property that was known at that time, and he called this nuclein. And this is actually what we now know as nucleic acids. So he actually discovered this. And this is actually what we find in DNA is in full desoxyribonucleic acid. The nucleic actually refers to this here found by Michel already quite some time ago. And then here's some, you don't have to know all of these names and these dates, it's just to make it a story, let's say, how we got to know all of this information. So I won't ask you about that. Levine, what did he discover, for example? So Levine in 1909, so already more than 100 years ago, he already thought about a tetranucleotide hypothesis. So that DNA consists of these 4 basic building blocks, which I think is quite amazing, more than 100 years ago. Only the way how they were linked was wrong. So this was his model, and by now we know Watson and Crick, of course, are very famous. But this was actually already one idea of how that could look like and how the building blocks could be linked to each other. Here they are, Watson and Crick. So they actually discovered the helical structure of the DNA, and they also got the Nobel Prize for it, and everybody knows them as Watson and Crick, also the Watson and Crick base pairing, but actually they didn't discover it all by themselves. I don't know if you know that. There are also credits that go to Wilkins and actually also Franklin. Rosalind Franklin shown here, which actually not really got credits for that. She actually took the X-ray diffraction image that led to the discovery of the helical structure. So Rosalind Franklin, but Watson, Crick and Wilkins got the Nobel Prize for the typical double helical structure of DNA. Now that's actually only one form of DNA. I don't know if you know that there are actually 3 forms of DNA. So you have aDNA shown here, you have bDNA and you have zDNA. what do you think is the form that you know? The B. So this is like, let's say, the normal form, but you can also actually have the A form or the Z form, but it looks a bit wrong, you could say. So what is different is, for example, here the grooves are different, and the way they turn and are composed are a bit different, and this is also discussed here. So again, we have here right-handed form, and then the ZDNA is actually left-handed. And this is how it all looks like. So I don't know, maybe first to show you another picture here. This, I think, is a very nice picture that shows you the different parts of the structure. So this is what we call the sugar phosphate backbone. So remember the backbone, we also had in the proteins. Here you see the sugar, here you see the phosphate. These are connected. And then on the inside of the helix, you have the ACGT, that of course everyone knows. If you want to look in more detail, then you can look at this here. So this is what we call the nucleoside here. Here the sugar, there you then have the base, and here you have the glycosidic bond. So again, this bond already can turn, like we have the Phi and the Psi in the proteins. This glycosidic bond can also turn. That will be one feature, for example, for the DNA RNA structures. And then what determines if it's DNA or RNA, and of course here on the 2 accent position, if it is ribose you have as a sugar OH group, or only the hydrogen, then you have deoxyribose. So this depends which sugar it is that it makes it DNA or RNA. And then you can have different kinds of bases attached to that, and this will of course determine then if it is adenosine, cytidine, guanosine, and thymidine. So this is for the DNA, and then of course you know that the T is actually replaced by the U, so the uridine in the RNA molecules. Again, here I won't ask you to draw them. So this is maybe to see the difference between the purines and the pyrimidines. The way I always remember which one it is, maybe a trick, is you have the Y here. I always think that you have like the Y form here. This is how I remember that as a trick. Okay. So then this is how the sugar looks like, and this is then important on the next slides for the numbering. Important to know if I talk about C1 accent or if I talk about C5 accent, these are actually the positions what I refer to. So that's probably difficult and quite impressive on some of the next slides where you will see that. But you can just have a look at this figure. So these full lines are actually showing towards us, and then the dashed line are actually inside the board, you could say, if you would look at it from a 3D perspective. So on the C1 accent position here, we have our base connected. So here on the inside, then here on the C3 accent position, we can have here in our backbone the connection with the phosphate, and also here then, so the C4 position is here, and then the C5 position is there. So which means on these positions, we can make the link then with the phosphates to have our backbone structure. So I already told you about the glycosidic bond. So this is one bond you can have, but also on the inside of your sugar you can have bonds. So also here. So remember we had the 2 planes for the amino acids then, but here this is actually, you will see that it's either, let's say, an N form or a Z form, as we will call it. And this is the hydrogen bonding here between them, so either 2 or 3, depending on which one you have here, which base you have. Then you can have minor grooves, major grooves. Again, the same representation. This is the building blocks, the bases on the inside, the phosphate backbone, which means these are actually the sugar and the phosphate. And then this here, maybe again as an example. Now to compare all of these and to define the structures, they made the Cambridge Accord to try to describe these structures. And you have two kinds of parameters that they will describe here, and it depends if you have complementary bases and base pairs or consecutive base pairs to take into account. So if there are complementary bases in the base pair, it means intra, which means here two next to each other. Okay, so which means one base, a second base. So what you can have is you can go in the three x, y, z directions. You can have them certain distance between them, right? So between two bases. These features. These features here are consecutive base pairs. So one base pair here and the next base pair, consecutive. And again, you can have the same one between these base pairs. Again, the same directions here, these features. And they then typically use these Greek letters, so if you want to refresh your Greek, this is just an informative slide to help you. Yes. Now, what was the problem at the Cambridge Accord? They did not define or agree actually on how they will calculate, and they did not have, let's say, a standard coordinate system. So if you would have it shown here, where is actually the point where you start measuring? So the coordinate system. That's why after this they had a second accord, which called the Tsukuba Accord. And in the Tsukuba Accord they actually determined the standard coordinate reference. And this is actually represented here. So how this works is, the x-axis is directed towards the major groove, this here. So you have to think this is actually where the helix is, here on this side, and then turning. So this is the x-axis. When the y-axis is directed toward the strand, so this is where the two strands are, so that's why it's directed in this way. So that's the y-axis. And then along this axis of the base pair C8, C6, this is the C8 here and C6 there. There you have the line. And then this is parallel to the C1 accent, C1 accent. This is the C1 accent, this is C1 accent. And you indeed see that this is parallel here. So this is how you have the X, Y. And then obviously, where is the Z? It's just here pointing in and outside. This is then where you have the z-axis, which means in the 5-axis, 3-axis direction of the strand. Because these are the strands, then you have 5-axis, 3-axis, and then 5-axis, 3-axis in the other direction. So this is then the z-axis. And then you have the central position, the origin, shown here by the red dot. And then if everybody uses this same reference coordinate frame, then we are all talking the same language, let's say, to describe how these nucleic acid structures look like. Okay, so I already told you about the glycosidic bonds here. What are the other bonds that you can have inside here? The sugar. The sugar ring, we call this in English puckering. Okay, so you can actually have 5 here: tau 0, tau 1, tau 2, tau 3, tau 4. And then maybe first I'll show you the ends, because in total you can actually have even more angles. So this is actually the complete figure of all the angles you can have. So remember, glycosidic bonds here between your base attached here. This is your sugar ring, where you have those 5, and then additionally you can also have here in your sugar phosphate backbone, you can also have angles here. Also again, alpha, beta, gamma, and so on. So these are actually all the angles, the possible angles you can have and can describe and measure in these DNA structures. Okay, so then let me go back. If we have a look at the conformations in nature, we only see a few ones appearing. So again, like with the proteins and amino acid angles, the Fibonacci angles, you can't have all possible conformations. Seems to be limited to a few ones. And this is actually described here. So two that you can have what we call the anti, and you can have the syn conformation, depending how it is positioned, shown here. And then the other important slide is, I already skipped it, I think, is here. I already mentioned that the S conformation and the N conformation. S south and north. Why? If you look here, this looks like the S letter, right? You can see that, the S letter. So this is what we call the S conformation. Or the C2 accent endo family. Why C2 accent? Because here you have the largest deviation on the C2 accent position of your sugar. The N conformation looks like this. This looks like the N letter, right? The N or the north conformation is the C3 accent endo family. You have the largest deviation on your C3 accent position here. So these are these two, N and south conformation. So C2 accent endo and C3 accent endo. So these are some of the possible conformations you can actually have. And okay, what do you want to tell more about that? So then here you have your phosphates attached to it on these positions here that I already told you. And then the other additional torsion angle 6 on the phosphodiester backbone is actually shown in this figure. Now remember, how did we call the plot we can make for the proteins, for the amino acid, the Phi-Psi angle? The Ramachandra plot? Yes, right? So we can have those two angles. Now, how could we do it for all of these angles to have a look at that? Any idea? Might already have looked. And we can use a conformation wheel, and that's actually shown here. So the Greek letters that you see here are actually those angles. Okay? So all of these angles are then shown there, and these are then the preferred conformations shown here, the regions. So if we would look at DNA molecules, we would measure all of those angles. This is where in this region we would find them. So 0, 90, 180, 270 degrees. If we would measure all of that and plot it in this conformation wheel, then you would have it in these regions here. Okay. And then here a bit of differences between the different A, P and Z DNA forms. How many base pairs, for example, and a turn, that is different, and then the positions between the nucleotides can be different. So for this then actually can use these parameters to describe that. Yes, so that I think is more or less the basic things of the DNA that you have to know. And then something about the interactions. So you can actually have 3 major types of interactions. And the first one probably already rings a bell. Ethidium bromide. Why? For what do you use that? Ethidium bromide, or did we used to?
Shell staining.
Shell staining, indeed. But why then did we not use it anymore? When I was in the lab, it's already quite some time ago, we used ethidium bromide, but now probably have cyber. It is still okay. But I think I've heard about cyber green and all of these other ones. So why wouldn't you use it anymore? Or at least why would you use gloves? Actually, the reason is because it is intercalates with DNA. So it could also do that with your DNA, and that could then why they claim is that it can cause cancer. I think that's the reason. So ethidium bromide can actually intercalate with DNA, and this was actually already found in 1975. So then you can see that the planar aromatic molecule can actually bind between the base pairs in the center of the DNA, shown here. Another one then on the right side is the daunomycin. This is actually a chemotherapy drug, which can be used to treat certain forms of cancer. It is also an intercalating molecule. So this is one kind of interaction you can have, the intercalation. A second one is the groove binders. So there are different ways. Of course, you have the small groove and the large groove. So you can have molecules binding in the small and in the large, so in the minor groove. So this is one such an example. If you want to visualize it yourself, you could use this PDB code and load it in your panel. So this is one example. Molecules in the major groove are less common, but you could, for example, here you see a local conformation where you actually have like a triple helical structure of DNA shown here. This is the PDB code if you want to have a look at this structure yourself. And then the third type of drug complex formation you can have, so an interaction with the DNA, is actually the DNA alkylation. And this is typically, here you see it represented, here the molecule. These alkylating agents, they covalently bind to the bases, and they are known, for example, as platinum drugs. So these are also used in the treatment in the clinic for cancer, these platinum drugs. So these are some examples of that. And then you can have also quadruplexes. So I already told you the double helix, of course. Then you have triple ones that you can have, but you can also have quadruplexes. Where do you find that? Typically here. So any idea what is this or why that could be important? Or what I think it does at the end of your chromosomes. So on your telomeres. So I think the older you get, the shorter these telomeres get. So this is where we can find such an example of tetrameric helices. And then typically, again, that I mentioned, is that you can hear at the center, it actually requires the presence of sodium or potassium ion in the center to stabilize that. That is what the red sphere here actually represents. So this was mainly on DNA, and then the rest, the smaller part, is about RNA molecules. You can have different kinds of RNA molecules. Of course, you probably know the messenger RNA, DNA, messenger RNA, and then you have your protein. But there are other forms. What other forms do you, for example, know? So you have yes. Other ones. How do they bring the amino acids to the typical structure then to make the proteins? Then you need transfer, RNA, tRNA. And then, of course, you have your ribosome, so you have, yes, indeed. rRNA or DNA is also possible. And of course, viruses are well known, so you have several kinds. This is what we call the Baltimore classification system, and these are then the kind of viruses you can have. So double-strand, single-strand DNA, and then also double-strand RNA, single-strand RNA is what you can have. And I think I just listed here some of the, I think what you know best viruses. So for example, for the flu is a single-stranded RNA virus, HIV is a retrovirus, hepatitis here, polio, rotavirus, parvovirus, and then the herpes, all the viruses you've heard of before. This is, I think, also a very nice structure. This is the structure of tRNA. And then this is the rRNA. So maybe the tRNA structure is what you could say is an L form. If you look at it like that, so L-shaped. You have two arms. This arm, this arm here. And then each colored part actually has a certain purpose, of which this is important here. The orange part is the 3-accent CCA tail. This is actually the binding site for the amino acid. So on the tRNA molecule, you'll have it here. So which means also for each amino acid that is being brought and built in your protein, you have a specific tRNA molecule. And then on the opposite side, what would you have there here? If you would have your amino acid here, what would you need? Of course, you need to read. So what would you have here? You see tip 3. 3 here, what would that match with? It's actually what we call the anticodon. So that actually matches with the codon of your sequence. Ribozymes are specific RNA molecules. You know enzymes. What is so specific about ribozymes? That they are actually catalytic. So they are catalytic RNA molecules. They can cut themselves or other RNA molecules. And we can actually have these classes of RNA or ribozymes. So catalytic RNA molecules. They can be self-splicing. They can be self-cleaving. Then you have the RNase P, and then you have, of course, the ribosomal RNA in the ribosome. And then some more information about that is shown here, how the splicing works, and specifically then this reaction. Then you have the self-cleaving RNAs. You don't have to know all of these, you only need to remember one. This is one example. If I would ask one example of a self-cleaving RNA, it's what we call the hammerhead. Why do we call it the hammerhead? If you have a look here, the hammer, like the head of the hammer. Yep. That's why we call it the hammerhead. And that, I think, if you would at least try to remember or could give one example of a self-cleaving RNA, is the hammerhead. And this is the RNA-P molecule, how it looks like. So this is the structure if you would want to load it yourself. And then maybe finally the ribosomal RNA, how that works. So this is actually a combination of proteins and RNA molecules. So I don't know if it's easy to see on the screen. So the blue ones here are protein molecules. And then you have the yellow and the orange are actually the RNA chains. So here and here shown. So this is what we call then the subunit, the 30S subunit in prokaryotes, and it's a bit different than in eukaryotes depending on how many proteins you have and how long the nucleotide chains are in the RNA molecules. This is actually how it works. Basically 3 sites. You have an acceptor site, you have a peptidylation site, and then you have the exit site where it leaves. So the first one is why the binding occurs, and the P site is the matching tRNA anticodon base pairs with a complementary codon. So here you see that it can read, so these 3 are, remember, the black anticodon matches here with the codon here of your messenger RNA. And then the last part, if it is released again, then you have the E site. So this is such an example. Now it's of course like a picture or a cartoon represented here, but this here is actually this molecule. So where you have your anticodon matching the codon, where you have your amino acid here, and then the other one is actually this complex of proteins and of RNA molecules, where you have the 2 ones, the large one and the small one. This is also then shown here. And this is then where you have your amino acids. So I hope that gives you a better picture of how it actually looks like. I think that's it for the second chapter. So this is maybe a bit more difficult than the first one, I think, also to study, especially with the puckering and the C2 accent, C3 accent endo. That is where most of the students think that's difficult. Now I don't ask into much detail, so you can have a look at the example exam. We already had the R exam for me, so I don't always ask into very much of the details. So that's important to know. Okay. And then the last chapter that I'll discuss today, maybe like 20 minutes, and then I will give a demo of the PyMol. Last half hour. So macromolecular structure determination. Do you remember the 3 techniques to determine a structure experimentally? For the first one you need Röntgen, same as when you break your leg, same method. So that's X-ray crystallography. So you have to make a crystal of your protein and then you just shoot X-rays on it and you can take an image, let's say. Second one is also in the hospital. MRI is the clinical application, so NMR, nuclear magnetic resonance. And the third one, if you have it where you work, no, you will have a normal light microscope probably, but not an electron microscope. So electron microscopy is also quite nice and yeah, I don't know. It's very expensive, I think, an electron microscope, especially now where they can render it in full 3D, make slices, then take the images and then render it in 3D. Things they have one at PIB. It's quite impressive. Okay, so this is what this chapter is about. The largest part is about X-ray crystallography. Why is this? Because actually most of the structures are X-ray structures. So I always tell the students that you have to know the order of magnitude, how many structures you have. So if you would have, let's say, in the range of 100,000, you could say that most of them are X-ray, then 10 times less you have NMR, and again 10 times less you have electron microscopy structures. So this is more or less the sizes. So this is actually X-ray to show you the difference. So here I think probably now we are at 150,000. In 2018 you will see NMR as less. So let's say 15,000. And then again 1,500, it's a bit more, let's say. So probably around 3,000 now. So not that many, I would say, for electron microscopy structures. Okay, so let's start with the first one. So the most widely used technique is the X-ray crystallography. This is actually the first image that was taken by Wilhelm Röntgen. It was a first medical X-ray of the hand of his wife. What do you think this is? The ring, yes. And actually, he got, again, I told you, lots of Nobel Prize winners. He actually got the first Nobel Prize in physics in 1901 for the X-ray technology.
Was niet doen in een gips, voordat ze een gips aan doen, was er ook iets en dan had ze een keer een kous aan en dat is ook op mijn
En dat staat er ook in.
Dat staat er ook op, dus er zit daar ergens iets in. Ik vind niet alleen denk van ik zou dat vragen, maar van die kousen dacht ik niet. Maar blijkbaar wel.
Misschien soms die haakjes zo aan vast.
Het was geen haakje,
De wind en dan zo'n haakje.
het was geen windel, het was gewoon een kous en een soort van...
Ja, vreemd.
Ja, stond er echt mooi in.
Okay, now why do we use the X-ray to visualize protein structures? So this is actually the electromagnetic spectrum that you see here. Here increasing frequency, increasing wavelength. I think you know the visible spectrum and the colors that we can see. Then you have the UV, and then this is the actually where you have the X-ray. And actually you have to look here, lambda in meters. How many meters then do we have? 10 to the power of minus 10. So you know meters, you know millimeters, you know micrometers. what is 1,000 times smaller, 10 power of minus 9 is nanometers. And then actually what is often used in protein structures and structural bioinformatics is Angstrom. You know, if you've heard of that, that's actually an A with, let's say, a dot on top of it, and that is actually 10 to the power of minus 10 meters. So these are the small distances actually in such a structure, and we need to visualize those distances, and this is actually why we use these X-ray technology actually to be able to measure that. And what we actually do is, you know, a prism, you have the bars on the windows. If you would have light shining inside of the cell, they would see actually the shades on the wall, right? The same principle is actually here, but only on a very smaller scale that you can't see. But you can actually then, if you have the proper equipment, you can actually take like an image of that. What do you need for that? We don't have the bars like in the window of the prism, but we can actually make a crystal lattice. We can make a crystal of our protein. And then on the crystal, we shoot the X-rays, and in a similar way, we can have what we call the diffraction pattern. This is, of course, the most simple crystal lattice, sodium chloride, represented here. Of course, for a protein crystal, it's more complex than that. But you could think of it like that. If you would shine light on that and you have it large, you would actually see on the wall, you would see the shades of everything, right? This is more or less the same principle. If you would do that in different angles, then you will actually know where each of the spheres are, the positions. So actually it's the same principle, but on a much smaller scale. And then, again, the name Max von Laue is actually who figured this out, that you could use that with the crystal. And again, he won the Nobel Prize for that, for the discovery of the diffraction of X-rays by crystals. So this is actually the picture that he could take. So this is the diffraction pattern from zinc blende, how it looks like. And then of course for a protein, if you would measure it, it's only much more complex. And then we need of course a computer and some mathematics to calculate the positions. And actually to know that we need what we call Brag's law. So I think this is the only equation that you will find in the course. You don't need to remember, I won't ask you the equation. But with this equation you can actually know then the distance and the position. So this is how the equation looks like. So you know the wavelength of your X-rays here, the angle, the distance, and by that, then here actually, if it diffracts here, then you can measure it here on a plate where it is and then calculate that actually. And for this, you can use Brag's law. The best known technique is single crystal X-ray diffraction, but there are other types. You have powder diffraction, fiber diffraction. This is actually then the process, how it goes. First of all, you have to make your crystal. This is quite a difficult thing to do. I made it when I was working in the lab. It took me a couple of years to get the crystal. Why? Protein, of course, you first have to clone the gene, produce it recombinantly in E. coli or maybe in another production system, produce quite a lot of it because you need milligrams of pure protein. So chromatography is the next step. You have to purify your protein. It has to be very, very pure. Like if you look on a gel, you only see one band of your protein. So all that process takes a lot of time, and then you have to make these crystals, and then you have to just test lots of different conditions, buffer, and then some additives you have to add to actually get to the crystal. And then when you have your crystal, what you do is you just shoot X-rays on it, and then it's gone again, you could say. So when you shoot the crystals on the crystal the X-rays, then you can get this diffraction pattern. So you already saw how it could look very simple here, but then of course for a real protein it will look much more complex actually. So as shown here. Once we have this, what we call diffraction pattern, then we can make or build our what we call the electron density map. So then we could know the position of our atoms and of our amino acids. This is actually where you see, depending on how good this went and how good you can see the distances, that is what will determine the resolution, then it will make it easier for you to fit in your amino acids and to have a high quality structure. That will come back later in the course. So then you have your electron density map, and from that actually you can try to fit in your amino acids, and then you can build your what we call atomic model. This is just one consecutive step, you could say. But then typically what you do is you try to refine your model, improve it. Just have a look. I found out this model. How well does it fit with my experimental data, with my diffraction pattern? And I try to improve it a bit and refine it until it can't get any better anymore. That's actually how you can remember it. So what we call the refinement. So these are the steps to get to the structure. So and most of the structures that you will look at during this course are normally X-ray structures and have been produced by this methodology. Okay, so I told you this is then how you build all of these proteins in a crystal, and you can have a crystal like that. This is actually the one that I made years ago. And there were actually two proteins, so it was a multimeric protein structure with two proteins, so you see the bands here on the gel. So what is important? Protein purity is important, also the concentration is important, also the pH is important, the temperature is important, and also the precipitants. You need these precipitants to have to get to the crystal actually. And this is why then you can have different techniques. First of all, one often used technique is the vapor diffusion. These are other possible techniques. And in the vapor diffusion, you have two options. You can go for the hanging drop. I think that's obvious. Here, the drop is hanging here, while here, the drop is sitting. Right? So these are actually how they are called. The hanging drop method and the sitting drop method. This is actually how it looks like. So you have here these crystal boxes. You have like these 96 well plates, but you also have 24 well plates. I think this is a 24 well plate then. What you do is you add a solution into that here. Your stock solution. And then here what you do is you add a drop of your protein that is purified in your buffer and your precipitants. And you add the same amount which is in the well. You mix that and you put that on top here of the cover. And then you just let it drop with a bit of grease that is sealed. That's important. And then you have something like this. So you make your drop here with a bit of this solution, you let it fall, and then you have your drop here. So hanging drop. Or sometimes people use the sitting drop method. So how does this work? This is, for example, how you could make it. So for the stock solution in the well, of course you need your water and then your buffer. What you typically do is you check different pHs. For in this example, condition A here could be then a certain amount of pH 7. Water, and of course, this is your precipitants. So ammonium sulfate in this case could be the precipitant, but sometimes other precipitants are being used. And then you just change the concentration a bit, and then hopefully in one of these wells, crystals will be grown. This is only one box. Often you have to make dozens of these boxes. You can choose if you want to make all of these buffers yourself, but of course, if you have lots of money, you can buy these kits where you have all of these different conditions already in Falcon tubes, and you can just use those to make these. Okay, how now can these crystals grow? For that, I think it's, well, I know the easiest way, but I'll try to explain. It's also something that students sometimes find difficult. Try to explain it based on this curve here and this plot. So you have undersaturation, and then you have supersaturation. Maybe an example, you could have, for example, let's say salt. You have water, you put salt in it, you mix it, it dissolves. Put more salt in it, mix, it dissolves. So, which means it's undersaturated. You could just keep on moving salt in it. Or you could do the same with sugar. Mix it, it will keep on dissolving. At a certain point, it gets saturated, right? This is then where we have this line here. Then we are at this point here. So it's the same when we grow these crystals. You have a certain amount of precipitants concentration and a certain amount of your protein. And the same way, at a certain point, you will reach saturation. Okay? If you just would keep on adding it, then of course you will get higher into this region, and finally, of course, it would precipitate. This is not what you want, of course, that your protein precipitates. Okay. So you have the solubility curve, and then the two areas undersaturated, supersaturated. I think that's clear. Then how do these crystals grow? They actually grow in the supersaturated solutions. So actually in this region here. Yes. So the supersaturated zone can be further divided into three zones. So the metastable zone here, nucleation zone, and precipitation zone. So of course this is not what you want. So you will try to work in these two zones here. This is what you try to do. In the label zone, the crystal nuclei are formed. So what they call nucleate, what we call here. So actually mini crystals are being formed. This is what you want at first. And then what you want them to grow. For that actually it's best that you are in the other one here. No nucleation, growth of nuclei. So the perfect situation then would be that you are just on top of this here, somewhere here. Which means that if it would drop a bit here, so you start getting this nucleation, and then it drops into this zone here, and then it just keeps on growing. If it would start here, you would just get lots of mini crystals, but they won't grow. That's more or less the idea. I hope that's more or less clear. So this is actually how we try to make these crystals. Of course they can grow nicely, but they can also have like these kind of defects. Things you can do, you can also seed. If you have certain crystals, you can actually not smash them, but actually divide them into pieces and then seed them and hope. For example, if you would seed them and they are in this condition, the seedlings that would only grow. So that's a trick that sometimes is being used. There are different ways of seeding them. You can start with large crystals and using these as seedlings. You can use microcrystals, or you can also do streak seeding. This is typically with a horsetail or a cat whisker of the cat that you use as a seeding wand. Why? It has to do actually with the structure of the horsehair or the cat whisker. That is perfectly actually to use it for the streaking of the crystals. And then of course you wait and you see if crystals grow or not, and then these are some of the views that you can get. So this is light precipitants, heavy precipitants, not really what you want. Here you have a bit of crystal formation and precipitants forming. You can have needle-like crystals, but actually what you hope to get is more or less something like this. If you are here, you have microcrystals, so you more or less are very close. And then of course you try to get these type of crystals. Sometimes you can have something like this. This is not good, this is just contamination. Or something like this, you could say, wow, the crystals are very huge. No, it's because it wasn't sealed properly. probably then you get this kind of dehydration, and then you get this kind of weird shape. But this is actually what you want. And then you can take your crystal and measure it, so fire X-rays at it. Of course, you don't want to shoot and just break them. Let's say you want to try to maybe protect them, that you can collect as much data as possible. And for that, you actually flash cool them in liquid nitrogen. You just grab your crystal in a really, really small loop, and then flash cooling it in liquid nitrogen. Then you place it here, and then fire the X-rays. And then collect the data. There are two ways. You can have a smaller installation. Some labs have this. For example, in Ghent. This is from the website. I found this picture. So they have this one in Ghent. But if you really want to measure and have high quality data, what you do is you go to a synchrotron. I don't know if you've heard of this, the synchrotron. There's at least one synchrotron that you know. Which one do you probably know? The Higgs boson particle, maybe he came into the news. So the CERN, the Large Hadron Collider, that's actually the longest one in CERN. And actually, this is in a tunnel of 27 kilometers long, and actually represented here, where you can actually fire them into the circle, high quality, and then these are what we call beam lines, where you can measure. Where you can have the radiation here, for example, you can have the X-rays, and then actually measure. And these are actually being used 24 hours a day, 7 days a week. So you have to reserve time to measure. So it could be that your time is at night and you have that many hours to measure. That's possible. So one example is the Large Hadron Collider in CERN, but there are other ones in Grenoble, Hamburg, Trieste in Italy. You also have synchrotrons where you can measure. And actually they are being used for several kinds of experiments. And one of them is for the synchrotron radiation, for example. And you can also use it for measuring your crystals. So this is then that kind of image you get. And then actually each spot you have here, you have to index it. And based on that then, on this information with the computer, and with some mathematics, you can actually get to the electron density map. So the mathematical technique, just that you know the name of the technique, what they use to get to this is the Fourier transformation. Maybe you heard of it. And this is actually then how a crystal could look like. So remember the sodium chloride crystal, actually then this is one unit, cell unit of a protein, and then this is built like this in your crystal, and then you try to solve this. You can have different ways how these crystals are being formed, and they call that actually space groups. And the 14 possible 3D configurations you can have of the points that can have an orderly arrangement build up a crystal are what we call Bravais lattices, Bravais roosters. And there are 14 possible ones. There's one that you definitely know. The atomium in Brussels is actually a crystal of iron. So here you see, for example, the cubic form, the body-centered one, the face-centered one. So any idea which one atomium is? Yes, the body-centered. So yeah, just if you look at it, you have to turn your head a bit like that. Look like that. So you have your 4 at here, and then 1 in the middle. So this is how the atomium looks like. So body-centered cubic form of an iron crystal. Of course, magnified lots of times. So you actually have 7 crystal systems, or in total 14 Bravais lattices. So these are, for example, 3 lattices, but the crystal system is cubic. Okay, and then you can have the same. Then, of course, you can also have space groups, but you don't have to know that for me. Just remember that you also have space groups, 230, but if you can remember the crystal systems and the lattices, that's okay. So these are the other ones. You can also have tetragonal, hexagonal, rhombohedral, orthorhombic, monoclinic and triclinic. And actually there the angles are a bit different, or the sizes can be, of the edges can be different actually. So there you see of the A, B and C, and then the angles are alpha, beta and gamma that they measure. And these are always a bit different, and then of course if you have one here on the center or not, makes then the different lattice. This is just an example from a paper here, then how it would look like for a protein as an example, and here's some other ones from other papers. And you want to have a look again, you find some codes, PDB codes, and you want to have a look at the structure of the proteins here. Okay, and then further to build actually your model, there's a problem known as the phase problem to actually calculate and get to the electron density map and the structure or the model. And there are three ways you can try to know the phase, know the lost phase, multiple isomorphous replacement, then single or multi-wavelength anomalous diffraction, and then molecular replacement. Molecular replacement is the easiest one. It means there's already another structure available that you can use to build the new structure. So using a homologous protein structure actually. Molecular replacement. These other ones are actually tricks you use by, for example, for the multiple isomorphous replacement, you soak heavy atoms in the crystal solution. And then they can actually co-crystallize, and that helps you to solve it. The other one is by using selenomethionine. So you know methionine is a building block of an amino acid. What you can do is, you can actually incorporate selenomethionine in your protein structure and use that as a trick. This is done for the anomalous diffraction then. And then the third one is the molecular replacement, where already there is a homologous structure available that you can use. So those are the three ways that you can get to know this lost phase. And then actually the goal is to get to your atomic model. So then if this would be, let's say, your electron density map, like this, then of course you have to fit in your atoms and your amino acids. So then of course here I think it's quite easy to fit this in, but if this is not that precise, it could be a bit more difficult, and the angles then would be a bit different. So this is, for example, then 5 angstrom, 2.5 angstrom and 0.5 angstrom, which actually just means the resolution, how good you can see the distances, how small you can get to see. And it's obviously, if you can get as close as 0.5 angstrom, you can actually know the position of each individual atom. While here it is a bit difficult, although this is still okay. But of course here, if you have a structure of 5 angstrom, it's actually quite difficult to fit all the atoms and the amino acids. So this is then a less accurate structure, of course. This is just then explaining what are, let's say, good resolutions and what you can expect of the structure for each of these resolutions. So that was the first one. The X-ray crystallography is the largest part of this chapter. And then the NMR. Just drink a bit. I'm talking too much. I want to tell you too much. So the NMR spectroscopy, maybe just briefly the principle. So it uses nuclear magnetic resonance. So this is the medical imaging device, the MRI, that Isabel was talking about. You already went in MRI? I never did. Multiple times. Okay. So then you know what happens. So here now, it's not the electrons that we use, but it's the nuclei of the atoms that we use. So here again, you have the atom, the nucleus, then electrons, neutrons, and quarks. and it's actually the spinning that we use of the nuclei here. You can have up or down, and it depends on the atomic field that you have. So this is, let's say, without, and then if you would apply magnetic field, you can either have a spin up or a spin down. This actually just means in which state you are. And that actually can be measured, and then based on the result of that, so this is represented here, you apply a magnetic field, you get these states, and then actually you get a signal, and based on that you can have a typical profile, and it is possible to deduce then the amino acids actually from this profile absorption spectrum. So this is an example here, then also how it looks like. Just to inform you, you don't have to remember that this is the paper where I found this information. And then this is just about how you can produce your protein. You can just read and study that. Then they have different expression systems, typically used, for example, E. coli. But you can also have yeast or insect cells, so it depends if you have problems to have a protein that doesn't something can be that your protein is not functional, and it needs, for example, certain modifications. So it could be that you cannot use a bacterial system to produce it, and then you need another system. For example, yeast and insect cells then, or even human cell lines are an option to that eukaryotic expression systems. The advantage is that it's, of course, not that easy to have it in high throughput. If you would have a bacterial culture, you can actually grow liters of it and ferment it in a bioreactor. But if you would have to work with cell lines, it's not that obvious, of course, to produce protein recombinantly. Then you have to concentrate it in these tubes, and then you have pure, hopefully, and enough protein in a high concentration, and then this is actually the tube. It goes in to the NMR spectrophotometer, and then you can measure it. Now what you get there is you don't get one structure or one measurement actually. So normally from an X-ray structure you will only get one model. If you measure NMR, you get what we call conformers. So different kind of conformations, typically about 100. And then if you look into PDV and you visualize, you often see 10 or 20 of these conformers. And this is actually one such structure. So what you see is in the middle, it's more or less the same, the different positions of the amino acids and the atoms, but the most difference is actually on the surface that you see here. So here, for example, you see there's a big difference there. So for each conformer, it can be that the position is a bit different. That's the difference in visualizing NMR structures and X-ray structures. Okay, and then the last part is electron microscopy. Again, some Nobel Prize winners here. Ruska got a Nobel Prize, and then here also for cryo electron microscopy. This was for the first electron microscope. And then this is more or less like a timeline, just informative. You don't have to study this. A timeline, what of the discoveries and which images they actually could take with an electron microscope. And then actually what you have to remember from this chapter is, of course, the principle here, but also the different techniques you can have. So you can have a transmission electron microscope. So it's, the technique is a bit different. Also the magnification you can have. The other one then, this is how a TEM looks like. So you can see the resolution is a bit different than with the other techniques. This is just a movie if you want to know how it works. Then the second one is the scanning electron microscope. Also a movie how that works. And then the last one is actually a combination. Then you have both the scanning and the transmission electron microscope. So that's the third technique, how that works. And then of course, if you could take different slices and different ways to visualize, you could get, again with the Fourier transformation, you could get a 3D image and reconstruct the structure with your electron microscopy images. And this is then typically what you get of structure, and then you can fit in these proteins like this. So then you can see it from a larger level actually, so not on a molecule level, but actually also try to fit it in these structures here of your microscopy images. So this is of course more advanced equipment that costs lots of more, and of course it's not used that often as, for example, X-ray diffraction. That's why we only have like a couple of thousands of structures for now of EM, but hopefully in the future we will get more. And this is then just the latest use where we are now in electron microscopy as at the end, and then you can have actually quite amazing images of structures. Okay, that's it for the third part on structure determination. And then now I would like to give a demo how you can use Python if you don't have experience. You still know how it works? No? Okay. What is the important part is to get it installed. So I would prefer that you install it in Linux, in Fedora. Why? Sudo dnf install Pymol and you have it. It's also possible to install it in Windows, but it's not that easy. Okay? So please try to install it in Linux. That's the best way to do it. That's why you might already have noticed that I'm also working in Fedora. So let me go to the first workshop. Maybe one more thing to tell you. So there are, I think, in total 10 exercises, and I already have the solutions ready. So maybe to give you an example, I will already publish the first solution, and then next week I'll publish the other solutions. So you can also have a look at that. Each step to get to the solution is explained there. So you can do it graphically, which means just click on the right and do several steps, or you can actually also do it all with commands. And this is actually what is shown here. So the first example is actually the hemoglobin structure. PDB code is 1 BBB. And then this is actually each step. Fetch, remember, I should already show that. Fetch is just to grab it from the PDB on internet, get it into your Pymol, and then these are actually all the steps that you could do, and then you could get to this result. So you can see often it's not that difficult. You just have to think about it, what is the best way to get, of course, to this nice representation. And it takes a bit of training to do that. But actually the commands are not that difficult. Okay, and so let me start with workshop 1. So there's a small introduction about that you have different programs. This is actually John Kendrew with the first model of insulin. So this is how he had to do it. And now we can just do it on our laptop. This is then an example of one of the first computers, a very expensive computer to do that. But actually now, thanks to the gaming industry, we can just have this gaming computer and visualize it, not even a gaming computer. I see you probably have a heavy gaming computer there, one also with but most of the laptops today can just run Pymol. You don't even need a special graphical card. It can also work on a normal laptop, no problem. So quite impressive, at least I think. There are quite a lot of different software. You can find a full list here. If you look in publications, you have several options, but mostly I find that they use Pymol. So in the beginning that I gave this course, like 10 years ago, I gave 3 programs, but it was too much, then 2. Yazara, I think, and Pymol. Actually, if you know how to use Pymol, you can do most of the things with that program. And it's open source. So you can just easily install it in Linux. That's also, of course, an advantage. Okay, so install in Linux Fedora sudo dnf install pymol, and then you should get a graphical user interface like this. What is important is you have a menu here on top. This is where you have a command line. You also have a command line here. It doesn't matter where you type your commands. You can use both. This is the canvas, the image where you will see the structure. And this is also important. This is where you will see your objects. You can have one structure, but you can also look at 10 structures and even compare them. Or you can split up a structure in different layers. And then you will just see here different layers appearing. This is an advantage sometimes to visualize things. Because here you have also a graphical user interface, a graphical menu, with the A stands for the basic actions, S is to show. Show could be, I want a cartoon, remember the Yiddishes, or the arrows, or cylinders for the strands. Or you could choose sticks, or spheres, or surface, are all possible ways to show. But maybe you first show a cartoon, and then on top, the surface. But then of course, oh, I want to get back to my cartoon. And of course you need to hide your surface again to see the cartoon. Another trick is you can make the surface transparent. That's also possible, and you can see both of them. And sometimes you want to label, especially if you have a quaternary structure with several chains. At first you look at it, oh, I don't know where's the A chain, where's the B chain molecule, you don't know that. You can just quickly set on the labels and then know where the chain is. That's always a good trick to use. But for making an image, for example, I always turn off the labels and just add the labels in like PowerPoint. If you are presenting a structure, just make your structure, insert the image in Pymol and use the labels in PowerPoint. That's much easier and better than with the labeling itself. Else you have to spend lots of time to get the position right of the labels. And of course you want to use colors. So then you have the color menu. Okay. So that are the basic things. And then the commands. Maybe I'll just give you the overview. These are the commands you will have to remember. Not that much, I think. Fetch is grabbing the molecule from the internet and getting it into your Pymol. If you want to remove it again, you use the delete command. If you want to color command, you want to show sticks, show cartoon, show surface, you use the show command. You want to hide it again, the representation, you use the hide command. Sometimes you want to set, for example, transparency, you can use the set. But there's also another nice way to do it, I'll show you. Create is important if you want to work with different layers on the right side. For example, if you load a structure like hemoglobin, you will have 4 chains. Chain A, B, C, and D because it's a tetramer. Let's say you want to color them differently. Then you have to type in each time a long command. You could also just create an A chain layer, a B chain layer, and so on. Then on the right side you will have 4 layers for each chain. That makes it easier to work with. And especially on the exam, it's often better to work with these layers, which will make you come to the solution more quickly than have to making longer commands, combining things to get to the visualization. So I often prefer to create different layers. And then finally, of course, you want to make a nice figure. For that you have to use the ray tracing. Tracing just means that what the program actually does is it tries to put a spot on the figure, then you can also get shades and it gives you a more realistic 3D image because you can see shading and light. And that you don't normally have. With the ray tracing you have that. Okay. I don't know if you have Pymol installed. Yes? So let's have a look then. So first maybe this one here, fetch 3CIG. So let me start my Pymol. Fetch 3CIG. Who remembers which structure this is? It looks like a... Is it this one you get?
Nee, ons scherm is al direct weg.
That's strange. I'll have a look at it at the end. So this is, how do we call it? It's a horse chain. A horseshoe, sorry. A horseshoe. So it looks like a horseshoe. Remember, with Yiddishes and then strands here. You see a few other molecules here. First of all, you can use your left click and your right click. If you left click, you turn it like this. If you right click, you move it up and down to zoom in and zoom out. So zoom in, zoom out. And if you want to translate it into space, you click on your wheel and then normally you can move it like this. So for this, I would use a mouse. Students come, first lesson, they don't have it. It's really terrible to see them with their mouse pads trying to turn or do things with that. So you really need a mouse to work with that. And okay, let me go back to the presentation. And then you could choose, often I always use hide all because by default normally, well they changed apparently on the version, now I have the cartoon already. You used to get the sticks by default, of course you don't want that. So then I would use hide all, so here. Then I don't see anything anymore. At least that's what I think. But here I still see 3CIG, so it's still there. Then I can type show cartoon, and there it is again. Cartoon is only now for the protein. So the other molecules, I don't see them anymore. Because you can only visualize them in a stick or in sphere or something like that. But the cartoon is for the protein structure in this case. It's also possible for DNA, but not for like glucose or other kinds of sugars or things like that. Okay. If you want to color, let's say if I would type color red, then it's in red. Color blue. Often these are terrible looking colors. So like I prefer is I would try to click on the right on the C, on the color, and then you can choose the spectrum. Red, green. So for example, for red, what I like is fire brick. For blue, I like sky blue it is, I think. Yeah. So often this looks a bit better. Also, I don't like a black background and a presentation. So what you prefer is a white background. The easiest way to do it, you can do it with a command, but you won't remember that command. So the easiest way is just to go to display, on top, background, white. In one of the next workshops that you'll see, you can actually change this, how the program starts and already start with a white background. That's possible. So in display you can already set the background. What you can also show is the sequence. That's also interesting. That you can have the sequence. Why? You can actually click here in the sequence and select things. So for example, I could just say, let's say I want to select from residue 100 to 120. I could just select it here on top. And then I can also see that it's selected there. If I would want to color that region specifically in another color, I could just click here and the selection. So now you see, because I made a selection, I have a special new layer selection. Which means I could just click here and I would only change the color. So in this case, for example, let's say I would color the selection in red and maybe do something terrible, show spheres, something like that. Just to illustrate what you can do with that. And then it also helps you, you also see it here changing in color. So this is just to show you the different modes. Here you can show lines, sticks, spheres, surface, mesh, dots, ribbon and cartoon. But in most cases, what we use is cartoon, sticks, spheres, and surface. Those 4 ones. In 95% of the cases, I always use one of those 4. And also in figures and publications, normally you see sticks or spheres or surface or cartoon. And then here you have an exercise how you can do that, so you can try this at home. Fetch to get the structure, show cartoon, hide, show ribbon. So you just do each command consecutive, and then normally you should just get the result here. Important, you can combine. So you could combine, for example, chains, if you have different chains with plus. If you want to know, for now, where the different chains are, you can actually also have a look here. Here you will see an A. So the 3CIG and the A. If you would have multiple chains, if you would scroll here all to the right, you see now I only have 1. Although here I have other molecules. If you see, for example, it's fucose, I think. You could also just click here to have it and do something with it, for example. I could just also click here. So this is then a 3-letter code. On the PDB website, if you would want to know exactly what it means, you can look that up, but that's for later. So these are then other kind of molecules. The O's that you see here are actually oxygens of water molecules. So sometimes what you also see is all these O's, or it looks like zeros, these are actually oxygens of water molecules. So this is probably a high resolution structure, and that's why you see this. You also see here amino acids in gray. That's in the protein, but if you click on them, you don't see them. You see they don't change here. That's why they aren't available. So they didn't get any data, or they couldn't, in their model, visualize them. So that's why it can sometimes be that you can see amino acids, but it's not possible to see them in a structure. In most cases, typically you see it at the end and at the beginning of your structure. But it can also be if you have flexible parts, they don't have a stable structure, for example, maybe a longer loop somewhere in the middle, it's also possible that that's shown in gray and that you aren't able to have a look at that. And there's an example, actually, I think, of one of these horseshoes where there's a part missing. Okay, and I think that's most important. Then for the rest, you can just go through the presentation. There are other ways. Also interesting to know is RESI of residue, then the index number, so the position. In this case, from amino acid 10 to 20. That's a way you can use. You can also use the name, RESN, residue name. Of course, unless you want to select all arginines or certain charged residues, for example. In most cases, why you use the residue name is for a specific molecule. For example, in the structure of hemoglobin, the example, I think there's carbon monoxide, that's CMO. Either you could click in the menu here, then to select that, let's say what I would do is for the fucose here, FUC, I could click on it, but I could also select it with residue name. So instead of RESI, I would use RESN, residue name, white space, and then FUC, fucose. And then I could also select the fucose molecule, all the fucose molecules. For example, in hemoglobin, if I select RESN, residue name CMO, there will be 4. Why? Because there are 4 hemoglobin chains, so there will be 4 CMOs in the structure. Also zinc molecules, ZN, residue name, ZN to select zinc molecules, and so on. Other examples. Any other important things I want to mention? So I showed you in display for the sequence and for the background. I think one more important thing is this one. The set, there are lots of options. I also don't know all of these options. I know some, sphere, scale, transparency, some other ones, but there are that much that you can't all know them by heart. It's actually easy just to look them up. So if you want to change a setting instead of using set and then the parameter here, what you can also do is just go on top here to edit all in the setting menu. So here, setting, edit all, and then I could just look here. So let's say I would want to use for helix, and I can see here cartoon, helix radius, round helix, all the parameters. If I want something transparency, I can have here all the options for transparency, and so on. For spheres, if I want to change spheres, I type in sphere, and then I see all the options here. So you can just actually click here and change a certain value. That's often easier than just set and then typing in the command. Much easier to find it. Another option is, there's a very, very good PyMol wiki. So Wikipedia only for PyMol. If you just Google PyMol wiki, this is the place to look. And in most cases, if there's a certain setting that you need for the figure on the exam, I try to fit in one special setting, either to look it up in the settings or on the PyMol wiki to get the maximum of the points. So that's already something that I can tell you. And then I think you know all the things, and it's up to you to practice. So try to make the exercises, try to look onto the PyMol wiki for a certain setting, and then hopefully you can get to the solution. But then next week, each week I will publish new solutions for the 10 exercises. There were a total of 4 workshops, I think, on molecular visualization that you will find in the modules on the learning environment. The last one is a very simple one, also the ray command, ray, and then 2 numbers here are just the resolution. So 800 pixels on 600 pixels. If you want a high resolution, it will take some time on your computer to calculate. So if you would say, let's say, ray 3000 on 2000 pixels, you will see that your computer needs to think about it and calculate to get the ray tracing and the shading and so right. Yes, there's one more very important thing to end the first contact moment is if you want to save it. Very important. Try to save it in between the exercises, also on the exam. Why? There's no undo in PyMol. You can see, unless they fixed it by now, but last year they didn't work the undo. So you might want to check it, but try to save it. It could also that it stops working, for example, so it's good to save it. How do you save it? You don't save it as a PDB. So the default, when you do the fetch from the internet, you get a PDB file. These are just XYZ coordinates for each atom. There's no information about cartoon or surface or color or anything. If you save it in a PyMol session, then you get all the information. So then you could save as 1bbb.pse PyMol session. Next time you open it, it will exactly look the same as you left it last time. The exact same view, the exact same layers here, view, color, everything will be exactly the same. So that's important. And so you do that by going to File, Save Session As, and then you can choose on your computer. And next time, of course, you can do File Open, open the PyMol session again, and then you can continue working. That's it for today. Any more questions? So I will have a look. Is it working now or no? I'll have a look. Yeah. Okay. So I'll stop the presentation now. The recording. And I'll just have a look. You get an error message or?
Ja,
Aha, ik zie het al. Het is omdat je een virtual box werkt. Ja, gaat er hier zijn waarschijnlijk.
Dus wat is in deze stadie?
Omdat je een virtual box waarschijnlijk niet cliqueren en afsluiten, dan ga ik kijken. nog een paar zaken kunnen aanpassen. Als je workshop ook eens een keer afsluit. En dan een keer daar in de settings. Ja, en dan het is niet veel doorrijden aan instellingen. Kijken of dat we een beetje kunnen dan een keer overlopen. In de vast, ik weet niet precies waar het zit. Dat het goed is. Het zal een systeem of zo. De geur is ook goed. Acceleratie. Dus daar zijn computer. Dat zal het maximum ook. Probeer je dat in te schakelen, 3D-acceleratie. Doe maar 2D ook. Ja, oké. Als je het wil installeren onder Windows, kan je op de Firewall wiki staat de uitleg. Het staat mooi beschreven. In principe, als je de juiste stappen in de juiste volgorde volgt, dus gewoon Google Firewall wiki. Misschien zelfs Windows install, voilà, je kent het al. En dan gaat er hier stap voor stap staan dat je het moet doen. Dus je ziet, je moet dan dit volgen en dan werkt het ook. Maar je ziet, het is ietsje complexer, omdat je hem eigenlijk zelf moet bouwen, zeg maar. Dus het is wel open source, maar nog niet gecompiled. Dus je moet hier zelf een aantal stappen doorlopen om het geïnstalleerd te krijgen. En onder Fedora is het.
Ik had dat daarnet afgespeeld, dus ik geprobeerd, maar dan is het maar 30 dagen.
Nee. Als je hem rechtstreeks van firewall.org of zo is het zo download, is het inderdaad een trial. Wat je wel kan doen, is een educational versie daar aanvragen. Maar in feite, als je gewoon via pseudo-DNS kan installeren of... Ja, het zal zijn wat gevirtualiseerd dat waarschijnlijk grafisch niet in orde is dan. En welke fout krijg je? Dan kopieer ik een segmentation fault. Ja. En misschien Python wiki, ja, gewoon Python erbij. Ja.
Ja, oké.
Ja, dat is niet echt, nee. En jullie gebruiken het allemaal gevirtualiseerd. Ja. Dan denk ik dat de Windows-optie als de beste gaat zijn, omdat gevirtualiseerd AI... Kan wel een keer kijken, bij mij, ik heb ook VirtualBox, of dat ik het daar kan oplossen. En dan eventueel, maar anders zou ik het voorstellen om het onder Windows te doen. Ja. Dan denk ik dat ik er al een keer een filmpje van gemaakt. Als het een probleem is, moest ik maar zeggen, dan ga ik het gewoon een keer bij mij onder Windows installeren opnemen en dat je dan de stappen gewoon kan zien. Het is gewoon omdat je hier wat commando's ook moet doen, En dan met pip install en zo, als je hier niet het juiste invult, kan het soms wat problemen geven. Maar dat is ook een van de skills als bioinformaticus, dat voor dingen geïnstalleerd te krijgen. Dat is ideaal om dat te trainen. Als het niet lukt, stuur mij een berichtje.
Het is
Gewoon een Frans maken.
spijtig gewoon in de Linux-stukken, omdat dat dan wat gewoon wordt van er meer mee te doen.
Ja. Ik heb daar dual boot.
Ja.
Dus ik heb me naar de schijf met Windows wat kleiner gemaakt en dan Linux geïnstalleerd om zo te kunnen draaien dat ik alle kracht heb.
Ja, ik dacht in het verleden dat wij dat ook gewoon hier bij biotechnologie was dat denk ik, zie ik dat panel erbij zat, dat wij dat gewoon ook in Windows hadden gedraaid.
Ja, je kan het in Windows perfect doen. Ik zeg, het is moeilijker om te installeren. Terwijl als je doet, je hem install onder Linux en het werkt.
Ja, dat is waar. Ja, zo moet het werken.
Maar ja, ik weet niet, als dat wel lukt, zo bij processen iedereen